All right, guys, welcome to this session. Sorry, my microphone might be a bit loud there. Welcome to the session, everybody. I can see how many people have we got in the conversation today. 18. Jeez, we're breaking actual semester records here. And the reason for that is we've got some therapy students joining us. So welcome to the session today, guys. I totally appreciate for some of you this might be a bit weird because some of your other lecturers may not present in this way, but this is just how we roll on this particular unit. And we're going to be talking about protein feeding and metabolism. But the most important thing that we need to do first is to say hello to everybody who has said hello so far. So Ryan kicked things off with a solid good afternoon, Mike. Thanks for joining us again, Ryan. We got Leona coming in. Leona, I'm not aware of you. I don't think we've met before, so thanks very much for joining and saying hello. We've also got Zara in the chat as well. Zara's been a staple throughout the semester this year, so that's absolutely fantastic. For the students who are usually on this unit, could someone in the chat just let me know that you can hear me, you can see me, and everything is nice and clear, just so we can head off any problems before we get into the session this afternoon. And whilst you're all doing that, for those that haven't joined you, you can see here that I have this chat box open. This is always open throughout the entire chat today, and that's the benefit of doing things live, is that if you have a question, I'll always refer back to the chat box, so you guys can submit your questions throughout. You can post them at any point, and then I'll just come back to the chat whenever something's coming in. If you feel you've got something to add to somebody else's conversation or a question that they've asked, that's absolutely fine. Just feel free to respond to them in the chat, and we'll just kind of work through it as we go from there. I can see Connor's come into the chat as well. Thanks for joining us this afternoon, Connor. Always great to have you all on board. So, excuse me, sorry. Two things first. I'm going to get into a bit of a description about what it is we're going to be covering on the session today. But the other thing is just to be aware, I am actually sat in a shed at the end of the garden. It's blazing sunshine outside, and I have the door open. So if you hear lawnmowers going off, or you hear children playing, there's nothing I can do about it, and I apologise in advance. If there's anything that's like horrific, then I'll just shut the door. But if I shut the door, it gets really warm in here, and yeah, I start sweating. It's not a good strong look for a YouTube based aesthetic, if you like. So yeah, just to bear that in mind, really. So if anybody else is prepared to stick their head up and say hello, that would be fantastic, because I can see how many people have we got in the conversation so far. Let's just have a quick look. So we got 19 viewers today. Fantastic, guys. So a lot of shy people in the conversation today, but that's fine. And my computer's having a meltdown. Right, let's get into it then. What are we going to be covering today? So we're going to be thinking about protein nutrition. Now, I know the therapy guys are interested in this from a kind of recovery perspective, and I can certainly try and make this a little bit more specific to you guys as well, because what we tend to do is look at what we need at baseline. So if you're just a completely sedentary individual, doesn't do any physical activity, and then we kind of look at it where we can start thinking about how we optimize protein ingestion. And again, now that I know that you guys have joined us, I can always have that in the back of my mind to sort of think about exactly how we can make that as specific as possible to you as well, so that you can make it a little bit more real in your world. We're going to think a little bit about the basics of protein metabolism and its integration, and I'm not going to take your head off too much with this, but there's just some real sort of fundamental stuff that we need to get out of the way so that your understanding is good, so that all of the stuff that follows that is in place and is in line. We're going to think about some of the 5 kind of core components of protein feeding, and that's pattern, timing, source, dose, and co-ingestion. And we'll talk about each of these different factors individually, but it will allow you to think about protein feeding in a slightly more structured and kind of logical way, so that when you start to provide advice or guidance to an individual, or you start to interpret kind of guidance from other places, then you can kind of approach it with that kind of 5 criteria kind of framework, so that you can then think about, well, does that make sense? Does that align with the literature? All that kind of stuff. And then we'll have a little look at the current recommendations and go from there. And then finally, how can we practically apply these recommendations? So is it realistic to say to someone, well, I want you to eat 3 grams per kilo per day for the next 6 months? Is that going to work? What does that look like in terms of food? how does that kind of pan out? So with that in mind, I am just going to hop across to a different view, just so you guys are aware that if you ask me any questions, that they will pop up on the screen and I can definitely address them as we go through. And again, from your sort of perspective, it just gives us the opportunity to just ask some questions where needed, and I can always refer back to it. So you can see Cheryl's, God, I always get nervous when staff come in. So Cheryl, thanks for joining us. Hi Mike, checking on the therapy students, looking forward to listening in too. Thanks very much for joining us, Cheryl. It's great to have you on. We've got Debbie, hello Mike. We've got Shaq again. If you use a personal Gmail account, I will use your personal Gmail name. So yeah, we'll go with Shaq. That's fantastic. We've got Leona. Good afternoon, Cheryl. How are you keeping? And we've got Ibert as well. Thanks very much, guys, for joining. And again, I will constantly refer back to the chat throughout the session as we move on today. So let's get back into our discussion and we'll go from there. Okay. So protein in the big picture, we're going to kind of think about, well, why is protein important within the human diet? So proteins are made out of amino acids, which are the building blocks of protein in the body. So without amino acids, those kind of core components of proteins, then we don't get proteins. And the amino acids will be unique to the protein because we'll get the kind of primary structure, the secondary structure, and the tertiary structure. And the tertiary structure is exactly how those amino acids all kind of fold together and interact with one another to form an active site. And of that active site, that permits that protein to do a specific function. And that's why protein is kind of different from the other macronutrients in the sense that we don't naturally have a protein store in the body. So we can store fats in adipose tissue, and we can store carbohydrates in the form of glycogen within the muscle and in the liver. But with proteins, there's not necessarily a specific store where we can sort of dip into that. And the closest thing that we have is muscle mass, because muscle mass represents the most amount of protein that is contained within the body. But as you will know from all different perspectives, that we don't really want to start using the functional tissues that we have within the body for fuel provision, because if we're starting to utilize our muscle mass for a fuel source, then we're actually utilizing the exact thing that we want to then use for movement or action or for completing some form of work. And therefore it's kind of counterintuitive. We don't really want to be doing that. And therefore we've kind of evolved to sort of go, well, okay, I'm now running out of the main macronutrients I would utilize for fuel utilization. What else do I have? Because I still got to keep functioning. I've still got to keep my body working. We've got to keep our blood glucose levels maintained at a specific concentration, and we can't let those things slip. Otherwise, we will start to then have sort of body failure. So our brain won't be able to function. If our brain can't function, then it's kind of all kind of downhill from there, really. So the primary things that protein is involved is in growth and repair. So as a functional tissue, those proteins undertake a task within the body. And if we don't repair the proteins that are undertaking these specific tasks, whether that's transporting ions across a membrane, whether that's contractile functions like that of skeletal muscle, or whether that's just a signaling protein to indicate that, you must increase the amount of muscle mass, or that we've got a large glucose load coming in from the diet, which means that we now need to release insulin, and the insulin binds to the cell to say, well, I now need to take up that glucose so that it's not increasing and accumulating in the bloodstream. So those are the kind of things that proteins will do from a growth and repair perspective. So we're going to try and think about exactly, well, the repair element is kind of like the recommended nutrient intake. So what do we need on a day-to-day basis if we weren't doing any physical activity as an absolute minimum to ensure that we maintain normal bodily function? But at the same time, a lot of us want to increase the amount of muscle mass that we have, whether that's from improving sporting sort of performance, or whether that's rehabilitating a muscle that's been potentially disused for a number of days or weeks, as the case may be. And therefore, how can we get that muscle back up to its prior strength? So a lot of the research within protein feeding is focused in sports nutrition. So that would be building and maintaining muscle mass. And it's a good population to study in because they're quite homogenous. So these studies tend to focus on very specifically recruited individuals who are typically male. I will say that off the outset because of the the consistency that you can get with research participants that are male. It's not to say that that's the right thing that the research community should be doing, but that's what has happened to date, and there is a bit of a tendency for that to be changing and adapting over as we start to appreciate the sort of physiological differences between males and females. So that's becoming a sort of hot research topic at the moment, to try and get a little bit more specific information about females and how they respond to different nutritional and exercise interventions, rather than just kind of assuming that everything must be the same as a sort of young healthy male, which is not always the case, or rarely the case, as the case may be. So in sport, I've kind of made that obvious connection, but obviously if we have an increasing amount of power that we can generate, then we can increase the amount of power to mass ratio. So the functional mass, and it represents a greater proportion of our total bodily mass. And as a result, if we have more functional proteins and therefore more muscle mass that permits us to do a function, then we will get a better performance. And this is no more apparent than in weight dependent sports. Things like cycling, for example. If you're carrying your mass up a mountain in the Alps, then you would want the vast majority of that mass to be actually be contributing to propelling you up the hill. If you're carrying an excess amount of adipose tissue, then that's not contributing to the amount of force that you can generate, and therefore it's weight that you have to then carry. And that's why you tend to see like ultra endurance athletes, or endurance athletes generally, runners and cyclists in particular, they tend to be very, very lean. And that's because they are trying to maximize their power to mass ratio. And the only way that you can really do that is by really concentrating on maximizing the amount of functional mass that you have, but and minimizing the amount of non-functional mass that you have. And the other example I've given here is that it can be quite important in some aesthetic sports as well. So things like gymnastics, in judo, and in like horse riding or jockey specifically. Those are all kind of aesthetic sports, but also that there is an inherent link between the amount of mass that an individual has and the amount of and the subsequent performance that they could have. So in identifying interventions or strategies to aim to optimize skeletal muscle mass, or indeed maintain what you have, is obviously going to be beneficial to lots of different people. So let's just have a little nosy in the chat, just as a polite reminder. The reason I'm looking down here, guys, is because that's where my laptop screen is, so I can see where the other channels are. Okay, good stuff. And let's just bring it up on screen so I can actually read it. That should be helpful. Good stuff, guys. So we've got just a little bit of chat going on there. So we've got Mihaela. Apologies if I've not pronounced that correct. We've got Nadia, Justin, Jessica, and we've got Jessica as well. Guys, fantastic to have you on board. Great to know who we're talking to today as well. And if anybody has any questions, then please do jump right in. That includes if you want me to repeat a point or if there's something that you didn't understand. So let's head back to the presentation and we'll carry on. Okay, so that leads me on to these 3 R's of protein nutrition. So we have the requirements. So how much protein that needs to be consumed for an individual to be in a nitrogen balance. We're going to talk about nitrogen balance in a second, and therefore avoid deficiency. We've got recommendations for each factor of protein nutrition to optimize skeletal muscle mass, and then the reality. So what do the athletes actually do in comparison to these requirements and the recommendations made, but then what do they actually then go on and actually undertake? So we know within the UK in particular that our UK reference nutrient intake for the general population is 0.7 grams per kilo of body mass per day. Now, hopefully all of you who are watching today are aware of exactly what your body mass is. Now, if you have your phone near you, or if you're on a computer, you're watching this, just open up the calculator, take your body mass and times it by 0.75 grams, and that will give you it will indicate how much protein it is that you would need to consume in order to reach or meet your reference nutrient intake for the general population in the UK. Now, the reference nutrient intake means that the vast majority of the population will meet their protein requirements if they consumed this amount. So that gives you a kind of rough idea of of what it is that you should be consuming on a day-to-day basis. Now, some of you will be undertaking diet records or weighing out foods, or have a really nice idea of exactly how much protein you are consuming in a day, and particularly if you're trying to target that to increase it? And if that's you, then let us know. Like, give us some of the numbers that you're sort of consuming, on what kind of frequency you've been doing that, and whether you find it easy to achieve that or if it's actually quite difficult. So if anybody has any reflections about their own protein consumption, then do share that in the chat, because it's always interesting to hear what people are doing. So then it leads us to the question, well, if that is for what the general population is doing, do athletes have a greater or lesser protein requirement than this? And it's a question that's kind of basic to answer, but quite complex to... it's basic to ask, but quite complex to answer. And it has a sort of different... There's been different opinions and different viewpoints on this throughout the literature and as it's kind of developed. So in increased amino acid utilization in athletic populations it occurs. So therefore requirements are sometimes less. However, increased requirements for greater tissue growth and repair and increased oxidation of amino acids are generally quite higher. They're much higher. And if you think about that from an endurance athlete who's doing lots and lots or thousands of small force repetitions, in terms of, like, if you're turning the pedals on a bike, you're contracting your legs thousands of times, so you're burning through lots of substrates, and therefore you tend to utilize or turn over more amino acids that are in the bloodstream, because they're then shuttled into the muscle and then utilized as a fuel, particularly during fasted state exercise or where feeding hasn't occurred for a prolonged period of time. Similarly, if you are trying to undertake a resistance exercise type approach, so you're in the gym, you're lifting heavy weights, then you'll have a greater range of damage, therefore a greater requirement for tissue repair, a greater signal sort of indicating that the muscle needs repairing, and therefore your requirements will go up. You'll oxidize more amino acids for the purposes of accruing muscle mass and repairing what's already there, and therefore that will drive up your requirement beyond that of the reference nutrient intake in the UK. So the current recommendations for endurance athletes tends to sit somewhere between 1.2 to 1.4 grams per kilo of body mass, and that for strength-based athletes is 1.6 to 1.7 grams per kilo. Now, I'm going to just hop over to the chat and see if anybody's come in and given me their protein intakes. Let's hope somebody has put something in. No one's answered the question. That's fine. But I just get you to reflect on if you've heard anybody talk about their protein intakes, and whether those numbers kind of align with your expectations of how much we should be consuming. And that's kind of an interesting reflection to have. And it's always, again, you can take your body mass and times it by these different figures to see what the types of intakes are going to be like for you. It's going to be a little bit out of context if you're not used to doing nutritional assessment, but for those that are used to seeing dietary intakes, you can kind of see that 1.6 to 1.7 grams per kilo of body mass per day is quite a lot of protein to consume, and it's not something that you could achieve passively. You would have to actively increase protein containing food sources in your diet in order to achieve that level of intake. So we've got Iber has just come in there. Thank you very much. Apologize for any pronunciations that I might be getting wrong. But after my body weight, I take like 105 a day protein. We don't know your weight, Iber. So what you could do is take that amount and divide it by your body weight, and that will give you the grams per kilo of body mass that you're consuming. And then if you just pop that into the chat, then at least we got a sort of rough idea of whether you're meeting your RNI, and that kind of 105 grams is comfortably going to be meeting your RNI. I'd be curious to know whether you're within this range from 1.2 to 1.7, but I can't answer that question because I don't have your body mass. So if you want to fill in the gaps, more than happy to do that. If you want to keep that information to yourself, that's absolutely fine as well. But I'll come back to the chat shortly, and we'll go from there. Okay, so let's head back over to the presentation. Okay, so I will be providing the therapy students with the slides for this particular presentation. I put it on the unit Blackboard for this, the unit Brio site for this particular unit, but I will share them with Cheryl, so don't stress about taking notes and all the rest of it. I'll send them through. But on this slide, it gives you a real sort of basic overview of exactly what types of roles. I mentioned them a little bit at the start, but what types of roles protein takes within within the body. Because sometimes when we think about, well, what exactly are these functions? Like, they're so far-reaching and so comprehensive that any function that your body undertakes, whether that's storing a memory, whether that's transporting a macronutrient across your gut, whether that's maintaining your nervous system, whether that's turning over the cells within a particular organ, all function is dictated by proteins within the body. And therefore, to sort of try and summarize exactly all of the functions that exist, we'd be here all day type thing. So I've tried to kind of summarize a lot of those here. So anything from sort of cell signaling receptors and signaling molecules, or structural membranes and enzymes that are embedded in those membranes, intercellular trafficking components. So when we move different components of the cell around. So one good example of that is the GLUT4 proteins that we use for uptaking glucose into muscle cells. They tend to be housed inside the cell, but then they get transported to the cell walls when either insulin is present or when we start exercise, particular endurance exercise. And that all of that function happens through the process of cellular signaling pathways, but also this kind of intercellular trafficking of components, and then creating these different little like lipid rafts that move to the outsides of cells and things like that. So ion pumps that maintain concentration gradients across ion channels, so your nervous system, for example, or the motor end plate for muscular contraction are also good examples. And then also oxygen and CO2 transporters. So if you think about the hemoglobin that's stored within your red blood cells, again, that is a protein. What else? There's protein in bones, muscles, tendons, again, hemoglobin, enzymes that catalyze all biological reactions, and then also used for repair and growth. So I can't stress the importance of proteins and the constituent components of amino acids. One thing that we haven't mentioned there that's kind of probably taken for a given that the nutrition students in the conversation today will know this, but we can synthesize proteins from amino acids, but essentially we don't have all of the amino acids so available to us, and there's some that we simply cannot synthesize from other precursors, and we call those the essential amino acids, because we cannot synthesize all of the amino acids that are available to us, and therefore if they're essential, what that means is they can only be obtained through the diet. So one thing to consider there when we think about amino acids and their availability. I'm going to head back over to the chat just to keep everybody awake and involved, and see if we've got any responses from the protein intake chat. Here we go. Okay, so I keep that information. I'm too big. That's absolutely fine. It's super personal information, so there's no issues on that front. Leona, my required protein intake is meant to be 134 grams, but I struggle to get that amount. I'd be curious to know how much it is that you do get, Leona, and what types of protein sources are you trying to target, if as sort of dwelling on your knowledge of what are high protein containing foods. So that would be nice to hear. Shaq, mine is 94.5 grams of protein intake. When you say that, Shaq, what do you mean? Is that your reference nutrient intake? Is that the amount that you would want to consume, or is that you know that you're consuming sort of 94-ish grams every day? So just let us know in the chat, and that'll be good to know from there. I'm going to continue on the conversation though, and we're going to move on with finding out a little bit more about proteins. So the digestion and absorption, we have a gastrointestinal tract here, and we've got a couple of different factors that we want to talk about with regards to the digestion and absorption. So it's pepsins and hydrochloric acids are kind of forming the vast majority of the breakdown that we want. So when we consume food, we're talking about complete and complex macronutrients. It's very rare, unless you're consuming amino acids that have been isolated and put into a powder form, like you can buy branched chain amino acids, or just individual types of amino acids like leucine and isoleucine and things like that. Unless you're just consuming a given amino acid, then it will be part of a more complex structure of a protein, found in animal proteins or in plant-based proteins as well. again, another reflection to think about it from your own sort of dietary intake is thinking about, well, do you have a vegetarian based diet or do you have an omnivorous diet? It'd be great to see what the kind of balance is like in the chat. So if you have the opportunity to share that information, that'd be really great, just to see what the balance is like. And therefore the different amino acid compositions within those two distinct sort of dietary intakes and all of the other different varieties that we've got, whether you're a pescatarian, ovarian, whether you're just a vegan or a vegetarian, what people's dietary intake is and how they categorize it is very unique and very specific to them, and therefore I don't tend to categorize people until we've had a kind of comprehensive conversation of exactly what they understand to be their dietary intake. But anyway, those proteins, from whatever source that they come into, will come into the stomach and will need to be degraded and broken down, and it's pepsin and hydrochloric acid within the stomach which is going to start that process. So it will denature the protein, it will start to unfold them and then start chopping them into their respective constituent amino acids. The pancreas releases sodium bicarbonate to sort of neutralize the hydrochloric acid. So you've got a very acidic environment within the stomach. You don't really want that traveling through your small intestine, so into your ileum and then into your small intestine as the main component. So therefore we kind of neutralize that. We provide a very alkaline solution called sodium bicarbonate, which neutralizes the hydrochloric acid, and it helps protect the small intestine. And then the small intestine can also release hormones such as secretin and CCK. Again, we're not going to get too bogged down into the exact process of this, but it helps to stimulate that digestive process further. It breaks down the proteins and continues to splice them into their individual amino acid components. And then secretin also helps to stimulate the pancreas to release more bicarbonate of soda to ensure that the acidic environment isn't maintained throughout the rest of the gastrointestinal tract. Let's just head back to the chat to see what the balance is on vegetarian versus meat source. I'd be curious to know what you guys have to say on that one. So Leona's nutrition shakes to give it a boost as I'm trying to shift a few kilos. That's interesting, but still don't manage it. That's absolutely fine. I've got no responses on the vegetarian and vegan front, or whether you're an omnivore or whatever. So if you just let us know in the chat what your dietary preference is. It'd be interesting to know, assuming you're happy to share that, and we'll go back from there. Please do answer the questions, guys, because it leaves me hanging. You know, it's like going for a high five and I'm just left there dangling. So Shamir has come in and said, Meat Man here, thank you very much for sharing that. So I'm assuming you eat a balanced diet, so no human being is going to allow to be just meat, because then you'll have some fairly negative toilet experiences doing that. But 3 portions of vegetables a day and 3 portions of fruit a day. That's my required intake from Shaq. Excellent, good stuff. So you've taken your body weight, times it by 0.75, and you've got 94.5 grams. That's fantastic. So I'm going to get in the sort of vibe of a vegetarian there, potentially even vegan, depending on whether you consume a more rounded diet. Nadia's come in at vegan. Excellent, good stuff. Leona, I eat meat but also eat tofu and falafels, and apparently are good sources of protein. Yep, there are high vegetarian containing sources of protein, and tofu is unique in that it has a complete amino acid profile, whereas a lot of vegetarian sources don't have a complete amino acid profile. Cheryl's come in a mixed diet, 25 to 30% meat, rest of the time mixed vegetarian and fish. Excellent, good stuff. Debbie's come in here and said meat defo, but I can eat lots of vegetables too. Excellent, good stuff. It's just interesting to get the kind of vibe. And it's, again, Cheryl, it's interesting you've categorized the sort of 25 to 30% meat and fish as being distinct entities, and a lot of people do this. I don't personally do that. They're just animal proteins. And again, most animal protein sources have a complete amino acid profile, which is the biggest differentiation we want to make between meat eaters and vegetarians and vegans. Again, if you're a vegetarian but you consume milk and cheese and dairy, for example, then the likelihood is you do get a nice rounded amino acid consumption because you're getting it from lots of different sources. So that's really interesting to know. And again, when you're thinking about an athlete who may be recovering and you've asked them a prior question, what's your habitual dietary intake? I'm a vegetarian or vegan, you may want to push them a little bit on that and sort of get a little bit of gauge for exactly how well they achieve their dietary intake. So is it complete? Are they eating different types of vegetables? Are they doing pulses, grains, different types of grains? Have they got a rounded nutritional intake? Because a lot of new vegetarians and new vegans actually struggle to get a rich and complete diet because they're only aware of specific types of foods that fit this new criteria, but they don't have the recipes, they don't have the cooking skills and things like that, and therefore they can actually become deficient quite quickly, particularly if they're only targeting the same sources all the time. Cheryl's come in, so approximately 60%, the rest is veg options. Yep, excellent. So Leonis, I probably need a nutritionist. Thank you for validating our entire career and direction. So yeah, great to know. I'm sort of being a bit daft, but yeah, it's interesting to see when you push people on some of these topics that additional information is sometimes required. Jessica's come in, meat but eat a lot of veg also. Okay, yeah. So hopefully the students that are on the nutrient metabolism kind of unit are also looking at these responses. I mean, I haven't seen any of my students yet, so guys, do feel free to chip in. But it's interesting to see the responses that we're getting and then the interpretation of diets and things like that. So it's actually quite nice to get some different perspectives, which is great. Okay, I'm going to head back over to the conversation or the presentation, sorry, and we're going to talk about a few different bits. So it's those freely available amino acids that we're kind of breaking down from these different protein sources, which we're then going to absorb into the bloodstream, and then we're going to transport them into cells across the body and create proteins from them. And if an amino acid is there in presence in excess, then there's, like I said, there's no protein store. There's no mechanism for kind of just popping that into a different compartment. And therefore, they're either converted into glucose or ketones. We're not going to go into ketogenic diets and things like that. It's interesting, but it's complex. And I'm just trying to, with my students, we've already sort of discussed some of this stuff. But for you guys, we're going to just stay on track with the protein stuff. But we can convert amino acids quite readily into glucose. And if they don't need the glucose, then they can be converted into ketones. Or it's quite simply, they're just decomposed, they're excreted from the body. So amino acid decomposition results in hydrocarbons and nitrogenous waste. Now, why, you might be thinking, well, why do I care about this? Well, proteins are the only macronutrients that contain nitrogen in them. And when we talked about nitrogen balance before, I said I'd make reference to this, then one easiest way of thinking about whether we are in protein balance or not is considering whether we are in nitrogen balance. And that's simply looking at the amount of nitrogen that we're consuming versus the amount of nitrogen that we're excreting. And it's a very basic way to think about exactly whether you're meeting your protein requirements or not. Because the only way that nitrogen is going to be present within your body is whether you've consumed it or not. And the only way that the nitrogen can be consumed is through protein sources. It's not going to be from carbohydrates, and it's not going to be from fats, because the molecular composition and the structure of those different molecules is completely different. It doesn't contain nitrogen. So looking at nitrogen balance is a very quick and easy way to think about whether you are in protein balance or not, or whether you're in nitrogen balance. And it's the urea cycle that processes nitrogen and facilitates its excretion from the body. And we're going to take a little whistle stop tour of the nitrogen, the urea cycle from here. So what we've got here, and I'm just going to use my little pointer here just to help show you where we're going. So we've got our intracellular protein source here, and we've got our amino acids. Now, the only way we have our intracellular protein sort of pool is from two places. From the dietary intake, that as in you've just consumed a high protein meal or a meal that's containing protein. It's quite difficult to not consume any protein, typically if you're eating a balanced diet. That then contributes to the intracellular protein pool, but so too does the breakdown of different proteins. So we're always constantly in this kind of cycle of synthesizing proteins and breaking them down again. That doesn't stop. It tends to go up and down depending on whether we eat it or not, because eating is quite a strong stimulus for synthesizing new tissue, but also periods of starvation are quite a strong stimulus for breaking down different tissues as well. So we can contribute to our intracellular protein pool from food sources, but also from just the amino acids that have been broken down from proteins that have either gone past their sell-by date, if you want to put it like that, or they're not functional anymore, so the body disposes of them. So therefore we have our amino acids as well. You see here, I've just sort of made a bit of an error. I would put an arrow to both of these, but I think we're just being pernickety here. But we have these freely circulating amino acids here, then go into these two distinct pathways here. So then we get our keto acids. If we're going, so again, just to be absolutely crystal clear here, we're not talking about protein synthesis here. We're talking about the metabolic fate of amino acids that have served their purpose. So we have two pathways here. We have the keto acids, which can go into the TCA cycle or the Krebs cycle at a very specific point. Again, I'm not going to get bogged down into exactly what point that is and what different of the sort of 5 different stages of the TCA cycle where that can occur, but the end point of the TCA cycle can be glucose if we are synthesizing new glucose molecules, or it can also be down towards respiration, so we're actually utilizing them as a fuel source, and therefore we can then obtain ATP and undertake some form of task with that. The other way is if we have an excess of amino acids, then we have the excretion pathway, and this is where we have our nitrogen and hydrogen containing component, and then we've got either the biosynthesis amino acids, nucleotides, and other biological amines which can go off and provide function for that, or they simply get excreted, and they tend to get excreted in the urine, and therefore if we took a urine sample, we could see exactly how much of those nitrogen containing molecules are going to be excreted into the urine. So the urea cycle is this kind of set of different biological or biochemical reactions, and it produces urea from ammonium ions in order to protect the body. So ammonium, again a nitrogen containing byproduct of the breakdown of proteins, is toxic. So if you were to consume it, it was poisonous for one, but if it accumulates in the body, it is toxic and poisonous, and therefore it needs to be excreted immediately. There's no kind of messing around there. It needs to be filtered out, usually in the liver, and then produced or converted into urea, and then disposed of out of the body via via urine. Again, I'm just trying to kind of adjust for the audience here a little bit, because I don't want to overwhelm people. But essentially, we have a cycle which we can undertake whereby these different amino acids and these different nitrogen containing components can be cycled round depending on what is being put into this cycle, and therefore what is going round and round it. So it's cyclical in nature. We can continue adding components to this cycle to ensure that those amino acids, and therefore that nitrogen, and therefore the ammonia, can therefore be excreted from the body via the urine. But again, we're not necessarily going to get too more bogged down from there. The other side of that, not the excretion pathway, but that kind of utilization for fuel. I get it. I think maybe I'll talk to Cheryl in future about what your prior knowledge is. I'm hoping some of this won't be too sort of alien to you. But if we think about how glucose is being utilized, we've got glycolysis into pyruvate, then we've got acetyl-CoA, which goes into our TCA cycle, and this is real kind of first year sort of biology kind of stuff, assuming you were exposed to that type of material. But this is the first pathway that you can undertake in order to generate and resynthesize ATP. We have to go through this kind of glycolysis into pyruvate, acetyl-CoA, and then through the TCA cycle, so we can accumulate lots of different electron carriers to go through to our electron transport chain in the mitochondria to then resynthesize ATP for muscle function. And what you can see here is that we've got a figure here where all of the different amino acids that we that can contribute to each of the different stages of this particular pathway. So you can see here that we've got these ketogenic amino acids, so phenylalanine, tyrosine, leucine, lysine, tryptophan, and isoleucine can all be converted into acetyl-CoA. Now, acetyl-CoA, when it's kind of bound together with another one, starts to form acetoacetate, and that's a ketone body. So you can see that these ketogenic amino acids are the precursors for binding two acetyl-CoAs together. This is a very simplified version of this, but it gives you a kind of rough idea. But out of the different stages of the TCA cycle, you can see where different amino acids are being injected into it that may have been excreted in the body. So if there's a requirement for this, then the body has a really good mechanism for actually accepting lots of amino acids in order to utilize them for fuel usage, so substrate usage. So thinking back to that kind of discussion we had a little bit about endurance athletes, whereby if they're exercising in a fasted state or they've been exercising for a prolonged period of time, then this can be a really good mechanism to ensure that they still have ATP being resynthesized so they can continue exercise. But it's usually at the detriment to your amino acid stores, and later down the line that can have a detrimental impact on the total amount of protein available, and therefore your muscle mass store in particular, because you'll start to utilize the functional mass that you have available to you in order to supply this particular chain. Don't think this is more of like a linking up slide. I'm probably not going to talk about it just now because it's a little bit more technical than we need, I think, for today. So we've talked about, how much do we need on a basic level? Does that differ for athletes? Yes, it does, by a sort of significant amount, and we've kind of reflected on exactly how much protein we all need for ourselves. So how do we then think about, well, if I was to change someone's protein consumption, or if we think about how effective a particular intervention is going to be, how could we go about assessing those different things? And we need to think about that from a kind of, is this a long-term intervention, or is it an acute intervention? Is it something we're doing over a number of months, or is it something that we're doing on a one-off basis, or even over a few days? And is it going to have a difference? What's the end point measure? What are we interested in achieving when we think about our particular intervention? So are we thinking about specifically about changes in muscle mass or the amount of muscle that's protein? Or, are we interested in measuring the actual amount of protein synthesis that's occurred within a given time window? Because we can do that. There's some really nice techniques available to allow us to do that. And therefore, how can we measure it? So if we're interested in muscle mass specifically, we can use things like DEXA, which is dual X-ray absorptometry, which is essentially using X-rays to scan both the bone mass, but also the the bone-free mass, so fat mass and muscle mass, either on a specific segment of the body or over the whole body, and that will give us our amount or our body composition in terms of grams or kilograms of a given tissue. So we can actually get from a DEXA scan kilograms of lean mass, and we can then segment the body for left arm, left leg, right leg, trunk, upper trunk, lower trunk, whatever it would be, to find out exactly how much muscle mass is present within those different areas. We've also got cross-sectional area for increases in lean body mass or muscle mass in a particular segment of the body. Or we can use stable isotopes, which is a specific technique for looking at how quickly and to what extent an amino acid is actually incorporated into a muscle, because we know that the amino acids are the baselines, are the base components for creating proteins. So if we could label an amino acid, we can then look at how much of that has been incorporated into the muscle itself if we took a little sample of muscle through a muscle biopsy, which sounds as horrific as it is. What are the issues? Well, there's some pretty expensive equipment going on here. So a DEXA scanner is going to set you back $100,000 or so, which is, is a lot of money. The control of long-term studies is really problematic because people are free-living beings, and therefore they tend to forget to do something, or they've got their own agenda. Like, I don't really like doing that, or I'm actually really hungry, I don't want to consume anymore, or I'd like to consume this, but it's not the intervention that you're doing. And therefore, trying to sort of balance the equipment needs and then the participants' ambitions and how long the study is and then how applicable all of the adjustments are that you had to make is actually really difficult. And therefore we have to kind of produce these really kind of intense but very well controlled short duration interventions to see if we can then extrapolate that out to think about exactly what it is that we want to achieve with our participants. So I've just said here, acute measures are not crucial endpoints. So we have limited extrapolation, but we have much easier to control. We can create environments and circumstances whereby we can have a lot more control over what we want our participants to do. Now, we are going to have a short break before 2 o'clock, and the session is going to run till about 2:30. So, and we've got quite a bit to cover, but there is some basics that I want to cover, and then everything after that is a big bonus. So when we think about net muscle protein balance, we can think about this in terms of nitrogen balance as well, but when we think about it in terms of muscle balance, we can think about it in terms of these two different components. Now we can think about it in terms of things that help to synthesize or create new muscle, and we can think about things that help to break down muscle, because as I say, we're always constantly in a state of either synthesis or breakdown. We're always constantly turning over the protein that we have available in our muscle on a day-to-day basis. Now, if our where's my mouse gone? Now, if our synthesis is greater than breakdown, then we have a net muscle protein balance. We are in positive muscle protein balance because we have more muscle protein that's accrued over a given period of time than has been broken down, and therefore we have a net muscle protein balance. If breakdown is greater than synthesis, then we're in a net loss. We've lost tissue, we've lost amino acid, we've lost functional protein over the given period of time that we've measured it for. And as a result, what we're trying to do is maximize the amount of synthesis that we get and minimize the amount of breakdown that we are experiencing. Because then if you have this period of time that's been repeated again and again and again, whereby we have synthesis that is greater than breakdown, then you will accrue more amount of muscle over a given period of time. And that's kind of the very basic principle around protein feeding in order to achieve muscle gain, or indeed the maintenance of muscle, that we try and aim for a greater amount of synthesis and we minimize the breakdown. And over a period of time, a number of months and indeed years, we will accrue muscle protein because we're in a positive muscle protein balance for a longer period of time. Now, what does that look like in terms of delivery to the cell? So we have a blood vessel, and this is carrying red blood cells, but it's also carrying amino acids if we've just eaten a meal. And then we've got our amino acid pool in the cell. And if we have a topped up amino acid pool, that means that we can synthesize new proteins and minimize the amount of breakdown that we've got. And there's this constant exchange of amino acids from the bloodstream into the cell, which is permitting the synthesis and also facilitating the breakdown. So collecting those amino acids, potentially recycling them, or indeed using them for substrate usage. But it just depends on the state of the individual. Okay, I'm going to head back over to the chat just to drop in with you guys and just double check everybody is okay and if there's any questions that have come in. Okay, I'm just trying to catch up just to see where we are. So Cheryl's coming. Is it an urban myth that some amino acids are only available in meat and therefore veg options require careful planning to be able to synthesize those amino acids? Yes, that is correct. So a lot of vegetarian sources or plant-based sources of protein have an incomplete amino acid profile, so they're incomplete proteins. So when you hear individuals make reference to complete protein sources, that is that they have all of the amino acids that are available to us, whether that's ones that we can synthesize or the essential amino acids. They're all represented in one protein source. It's that all animal-based proteins are complete protein sources just because of the very nature of the fact that they've come from an animal, and therefore they also need the complete amino acid profile. Whereas plants, because they have different metabolism, different biochemistry, they exclude certain amino acids, and therefore, like you say, unless you target a range of different protein sources from different plants or plant sources of protein, then you may fall into the trap of having a gap in your amino acid profile, and therefore you would become deficient in that particular amino acid. And therefore, like I said, I gave that example before of if you're a new vegetarian or vegan and you don't have the kind of cooking experience, or you don't have a full appreciation for the true breadth of foods that you can consume, then you can fall into this trap quite quickly and quite easily. Macy just come in. Isn't it the Krebs cycle? So the TCA cycle, the transcarbolytic acid cycle, the Krebs cycle, TCA, they're the same thing. They're just used interchangeably, and apologies for not making that differentiation. Shaq, yes, I will definitely be passing on the slides. Cheryl, we've covered energy systems, fantastic. So at least I know you guys, I wasn't talking a completely different language. Excellent. Perfect, guys. Any other questions that spring up, then just let me know and I will try and get to them in the chat today. Let me head back over to the slides and we will continue on. And I have scheduled a break, so if you're feeling a bit overwhelmed, I totally get that. I'll try and slow down and just make sure we're covering everything in the detail that we want it to. So, amino acid provision. What we've got here is a figure, and what we've got on the y-axis here is muscle protein balance, and you can see here we've got positive and we've got negative, and if we're in the middle, then we're neutral. We've not gained or lost any muscle protein or amino acids indeed over the time period of interest. Now, when we're in rest and we're in a fasted state, as in we've not consumed food for sort of 10 to 12 hours, then we're in a net muscle protein we're in a negative muscle protein balance. So we are at a loss at this particular moment in time. Now, what this particular individual has done, or this study has done, the Biolo studies here, is shown what is the impact of just giving someone some amino acids. So no exercise, but giving someone freely available amino acids in a sort of dietary supplement. We can then have a look exactly what happens to their muscle protein balance when they've had some amino acids. So funnily enough, if you increase the amount of amino acids available in someone's diet, then it increases the muscle protein balance into a positive muscle protein balance, because you have amino acids available in order to move into the cell, synthesize new proteins, and therefore increase the amount of muscle protein balance that you have. If you were to take resistance exercise, we see a slight blunting that we would get in comparison to rest, but we're still negative. And if you were to undertake resistance exercise and continue to be fasted, then you would exacerbate this issue. You would continue to be in a negative muscle protein balance because you have not replaced the amino acids that you may have utilized, but also you are not repairing the damage that's been done by undertaking resistance exercise. Now, the final combination that we're going to look at here is resistance exercise and the provision of amino acids. And this is where we see the biggest anabolic effect, and an anabolic effect is one of growth, of one of development, of accruing, because you've provided the muscle with the substrate that it needed, but also with a physiological stress in which to utilize those amino acids in the most positive way that it can. And that's what it has, this kind of accrual effect, or this kind of synergistic effect, whereby the resistance exercise provides a potent stimulus, and the amino acids are readily available to be utilized to increase the muscle protein balance of individuals who have undertaken resistance exercise and consumed amino acids. So amino acid provision increases the muscle protein synthesis, resulting in positive net muscle protein balance, which is kind of what we're wanting to achieve. That's what we want to do. So when coupled with resistance exercise, that amino acid provision results in greater positive net muscle protein balance. So resistance exercise and amino acid provision is the most potent anabolic stimulus that we can give to an individual. It doesn't get kind of much better than that unless you're starting to use a kind of pharmacological approach, which is generally frowned upon, particularly in performing athletes. So, with that in mind, now we know that resistance exercise and the provision of amino acids, or protein more specifically, we can then start to think about, well, that's quite generic advice. How can we manipulate the different criteria in order to optimize that particular response. And we've got these 5 different components here. So we have the pattern, the co-ingestion, the dose, the source, and the timing. And you can see that all of these factors overlap in the middle in order to rechange or alter our skeletal muscle protein remodeling. And what that essentially means is that each of these different factors contribute to the makeup of the amount and the makeup of the skeletal muscle that's present within your body. So when we talk about pattern, we're talking about over a 24-hour period, how frequently are you providing an individual with protein? So is it twice a day? Is it 3 times a day? Is it 5 times a day? Is it 8 times a day? What's optimal? Because that is intrinsically linked to the dose. So the dose is simply how much you would provide, how much protein you might provide to an individual over a given period of time. Now we've talked about the types of ranges that we want to try and achieve in terms of ingestion. But if your pattern is 8 times a day, but you've got a fixed amount of dose, then that's going to reduce the amount that you're consuming on each individual feeding opportunity. And is that optimal? And that's some of the things that we're going to unpick in the sort of second half of this presentation. The timing, so how closely to the exercise bout do you provide this particular protein source? Is it before? Is it during? Is it after? And if it's after, how long after do you get this kind of anabolic window? Does a window exist? We don't know. We're going to try and find out. The co-ingestion is an interesting question as well, because I'm talking about it in a really kind of controlled and basic way, talking about what protein sources can we give to an individual in order to increase the kind of synthetic response that we get. But is there actually any benefit to consuming it or co-ingesting it with another macronutrient, whether that's carbohydrates or fats? So we'll have a look at a little bit at the evidence behind that. And then finally, we'll think about the source, which we've kind of already talked about there, about the amino acid profiles, whether it's a complete or an incomplete protein source, and whether there are actually some specific amino acids which are really particularly potently anabolic. Do they actually drive that response more so than others? Now, I think I'm going to pause here because it's a good time to pause, and we are going to come back just after 2 o'clock. So that gives you sort of 6 minutes or so just to kind of stand up, stretch your legs, grab a cup of tea, and just have a bit of a breather. And then sort of on the hour, maybe just slightly after, I'll pick up the conversation, and we're going to start with dose as being the primary factor that we want to talk about. So I will see you guys in a few minutes time, and we'll pick up the conversation from there. See you shortly, guys. All right, guys, welcome back. It's a bit of a tradition throughout the semester for people to share what it is that they brought back to the table with them. Extra points for the most interesting thing that someone's brought back. My lot used to just bring water back, and I'm like, geez, that's not true. May's usually pretty good at bringing something interesting to the table. So if you're snacking on something particularly unique and fantastic, then do let us know in the chat. I went boring. I had a can of Diet Coke, which is, I know it's not on brand, but it's fine. It's all good. It just gives me that caffeinated boost that we require midway through an hour and a half's worth of session. So anyway, without further ado, whilst you're all telling me exactly what it is that you brought to the table, we're going to think about dose. So this is one of the components. We're going to kind of work progressively through the components, thinking about some of the evidence that there is for why you might take a particular approach when it comes to protein consumption. If there's anything that's not clear, I'm going to get through quite a lot of literature in not a lot of time because we're a little bit pushed on time today. But I'll try and keep it as basic and sort of fundamental principles as possible. And if there's any questions after that, then we can kind of fill in the gaps. So let's just get rid of me and make me a bit smaller. Fantastic. Right, so dose. There was a study done by Dan Moore et al. In 2011, and this was published in the American Journal of Clinical Nutrition. And we can see here that essentially Dan gave individuals a range of different amounts of protein. So you can see here we've got no protein at all. Then we've got 5, 10, 20 and 40 grams of a complete protein source, which I believe was whey in this particular example, which is a commonly consumed protein based nutritional supplement. And what we can see here is that the letters represent significant difference. So you can see here that A is different from B, and B is different from C, and C is different from B and A. So if the letter is different, it means that the groups are statistically distinct from one another. And what we can see here is that we have an increase to 5 grams, and then another increase to 10 grams, but not different from 5. And then we see this jump up to 20 grams. So 20 grams is distinct from 10, 5 and 0. But then when we get to 40 grams, a doubling of the amount of protein that we've consumed. So a double, 5 grams, a doubling, another doubling and another doubling. It doesn't result in a further increase in the amount of muscle protein synthesis that's taking place. So the unit here is muscle fractional synthetic rate, and this is a very tricky physiological technique that you can use to look at what amount of a given or known amount of amino acid is incorporated into the muscle. You take a muscle biopsy, then you can then detect exactly how much of that particular label is actually being incorporated into the muscle itself. So if you then control the amount that you've given to an individual and then sample them at specific time points after the point of consumption, then you can then start to actually calculate, well, what is the fractional synthetic rate of this particular amino acid over a given period of time. And what this study showed was that 20 grams of protein was sufficient to maximally stimulate muscle protein synthesis in comparison to even 40 grams. And 40 grams didn't result in any further increase in muscle protein synthesis, and this was consistent whether it was at rest or whether it was following resistance exercise. So you can see here a study done by Oliver Whittard in 2014 confirmed this. So you can see rested exercises in the white blocks and rested and feeding is in the white blocks, and exercised and feeding is in the black box. And you can see 0, 10, 20 and 40 grams of whey protein was consumed. And again, it looks like there was an increase in the mean between 20 and 40, but this was not statistically significant. So the variance here was too great in order to confirm that there was a further increase. So no longer a dose response relationship between the amount that you've consumed and the myofibrillar fractional synthetic rate, or the muscle, the amount of protein that you're accruing. So Dan went back to the table here, and my reference is sort of blurred out here, so apologies for that, because it's in white. I can fix that for when I send it round. But you can see here that Dan did a meta-analysis here and took lots of different studies. So each one of these circles represents different studies, and therefore we can then calculate the amount of protein that an individual has consumed in terms of grams of protein per kilogram of lean body mass. So these individuals will have had some form of body composition undertaken in order to determine exactly, well, what amount of protein were they getting per kilogram of lean body mass. And you can see here that there seems to be an inflection point here about 0.25 grams per kilo of lean body mass, particularly at rest, and therefore there seems to be this kind of diminishing returns where you see this plateau. When you look across lots of different studies, there seems to be a plateau in the amount of fractional synthetic rate that you can actually achieve when you feed. And if we think about 0.25 grams per kilo, then for a 100 kilo individual, that's 25 grams. So again, when you start to look at what that 0.25 grams represents, it's quite consistent with the other studies that have been previously published on this, whereby there seems to be diminishing returns with protein synthesis with any amount greater than 20 grams. So what are the other factors that may consider and influence this? And those are the things that we're going to go into. So it seems like 20 grams on a given feeding opportunity seems to be sufficient to maximally stimulate muscle protein synthesis. So that's one thing that we can think about: dose. On a one-off occasion, if we want to maximize muscle protein synthesis, then 20 grams seems to be the minimum we need in order to maximally stimulate the dose, the response that we're looking for. Which then led people to think about the pattern of this. So 20 grams, as you've seen through the calculation that you did, or sort of back of the envelope thing, where you took 0.75 times it by your body mass, no one will have got 20 grams, because you would have to be, well, I can't do the maths on that quickly enough, but like no one's like 5 or 10 kilos in weight, because you're not, infants, as it were. And as such, 20 grams is okay for a one-off feeding, but it's not going to meet your recommended nutrient intakes, and therefore it represents the optimal dose that you can do on a given opportunity throughout the day. But if you want to meet your recommended nutrient intake, then how frequently should you be feeding 20 grams? Now, you could just say, well, my recommended nutrient intake, or the amount that I want to consume, is 100 grams. So I'd need 5 opportunities to do that. And therefore, you can then evenly space out those 5 feeding opportunities and potentially maximize the dose. Whereas we know that when people consume food, we tend to have sort of a sort of small to moderate breakfast, a sort of medium-sized lunch, and then the vast majority of people in the UK will have a larger evening meal, and that evening meal will tend to have a higher protein, a higher protein amount than the other two feeding opportunities that we've had at breakfast and lunch. And again, if you incorporate snacks, most people will have a cereal-based snack, as in a grain or a wheat-based snack, which again has a protein element, but it's not the primary source of that. And that's what I'm talking about in terms of if you're trying to increase your protein consumption throughout the day, you would have to consciously go about doing that, and be aware of what those protein sources look like. And as such, that's what these investigators have done here. Now, one particular research group is Phil Atherton at the University of Northumbria. University of Nottingham, not Northumbria. And there we were interested in this idea that you would have to reach a certain threshold in terms of the amount of amino acids that are in your bloodstream in order to trigger a protein synthetic response. And that's what we can see in this figure here. And I'm just going to put my pointer on so you guys can see where I'm pointing. So what we've got here is just the y-axis is the data span. So the concentration of concentration of plasma leucine, but also the fractional synthetic rate, and it's expressed as percentages so that you can compare two distinct measures here on the same figure. And we can see here that around about 100 minutes after the time of consumption, we see a peak in the plasma leucine concentration. Leucine is an amino acid that's commonly found in things like whey protein, for example, and it's easy to measure as well. And then you've got fractional synthetic rate, and then the observant lot of you watching today will be able to see that there seems to be a correlation between the plasma leucine concentration and the maximal or peak fractional synthetic rate. And again, this is also consistent when you look at the intramuscular leucine concentration. So that's plasma in the bloodstream. Now this is in the muscle, and you can see Sorry, I thought my child had suddenly arrived. You can see here that the two track each other individually, quite specifically. So the fractional synthetic rate peaks at 100 minutes and then it drops quite severely after that. Now, what this led the researchers to think about was thinking about the fact that you see this quite distinct decline in fractional synthetic rate and amino acid provision in both the bloodstream and the intramuscular leucine concentration, which means that you don't necessarily have the leucine available or the amino acids available in order to stimulate fractional synthetic rate. So therefore, what this then led researchers to think about in terms of the pattern of feeding is that your amino acid provision must be sort of sufficiently far apart in order for you to allow repeated stimulation. So what that means is, if you think about this peak here, if you just go back a second, you would want this peak to occur. That we know that fractional synthetic rate occurs here, but then our plasma leucine concentration is still elevated here, but our fractional synthetic rate has dropped off. It's dropped off a cliff, almost down to baseline levels. So then your next feeding opportunity needs to be sufficiently far away so that you can then increase your plasma leucine concentration and then drive fractional synthetic rate again. It couldn't be right here because our fractional synthetic rate has dropped off sufficiently. So it needs to be far apart in order to repeatedly stimulate that particular response. And that's exactly what a bunch of researchers went on to try and understand. And the study by Oretta et al. In 2013, published in the Journal of Physiology, attempted to show this. So they did a bolus ingestion, an intermediate ingestion, and a pulse ingestion. And you can see in the text behind my head here that we've got the pulse would be 10 grams 8 times a day. The intermediate would be 20 grams times 4 times. And the bolus would be 2 40-gram ingestions. So in every single example here, they are consuming 20, 40, yeah, sorry, they're consuming I'm just double checking the maths there. They're consuming 80 grams in a 12-hour period, because some of you might think, well, that's not sufficient over a 24-hour period, and you would be correct, but this was only done over a 12-hour period. And they did they did a bilateral leg, so they looked at rest in one leg and rest and exercise in another leg, and they showed that the myofibrillar fractional synthetic rate was highest during that intermediate dose, which was significantly higher than the bolus and significantly higher than the pulse. And that's, again, kind of reinforcing that picture that you see with the amino acid provision, that 20 grams seems to be sufficient enough to drive up the amino acid concentration to reach that threshold required in order to start muscle protein synthesis. The pulse doesn't seem to be enough. There doesn't seem to be enough amino acid there to help fully stimulate that pathway. And similarly, the bolus one is provided as a really infrequent it's provided so infrequently that you're missing opportunities to make the most of the amino acids that have been provided. And we know that 40 grams is no more beneficial than 20 grams, and a lot of that will just simply be excreted. So therefore, 4 20-gram doses of protein over the 12-hour recovery period from resistance exercise was the most effective strategy to stimulate post-exercise muscle protein synthesis. So 20 grams on 4 different occasions over a 12-hour period seems to be a really good optimal feeding strategy in order to maximize the protein synthetic response from resistance exercise and feeding. So an even distribution of protein throughout the day results in a better MPS, average MPS, muscle protein synthesis over the 24-hour period. And again, you can see here that a further study by Mamaro et al. Sort of showed this by trying to control over a much longer period of time, that they either did even 30 grams of protein throughout the day from day 1 to day 7, or they did a skewed, so they did breakfast as 10 grams, lunch as 15 grams, and then dinner as 65 grams, very similar to what I described before in terms of a common UK diet, and showed that you saw a blunted muscle fractional synthetic rate when you skewed your protein consumption rather than trying to optimize each feeding opportunity from there. I'm just going to drop into the chat and see if anybody has asked any questions, because I know I am kind of rattling it off a little bit here. So let me just drop in. Good stuff. And why is it not coming up? Here it is. Perfect. So low engagement with the snacks. Leona, a glass of Pepsi Max, good for you. And Cheryl, a tea and a bit of my... Oh, Cheryl, I tell you, I'm saying it with shock, but my children got a lot of Easter eggs this year, and I felt like they needed some help. So I kind of know where you're coming from on that perspective. I've got a little bit more mass to put all that chocolate into, and even more mass as a result of that now, but never mind. Thank you for sharing that, guys. Again, any questions, just jump in and we'll go from there. We're going to move on from thinking from the dose and the pattern. We're going to now think about the timing and whether there's any evidence to suggest that the timing is important when we think about protein feeding. So you see these memes knocking about on the internet, and I'm just get my screen up so you guys can see that. Love a meme, so let's share them now. So just finished my workout, can't miss my anabolic window, and you can see that this lass is running towards getting her thing. We've also got our anabolic window, suggesting that some people don't believe that the anabolic window exists. And also, missed my anabolic window, workout was completely useless. So you've hit the gym, you didn't bring your protein source post-exercise. Is it all just a complete waste of time? Do you reap any of the benefits? Who knows? But in terms of meme generator here, our Starship Trooper is a little bit disappointed that he's missed his anabolic window. So what evidence is there that there is an anabolic window, or whether one exists? Now, this study by Cribbs and Hayes here looked at this kind of co-ingestion of a protein, creatine and carb supplement, and you can kind of see the details of exactly what they've given here. And they've tried to maximize this over a 10-week resistance training bout with 23 resistance trained individuals with 4 sessions per week. And they did DEXAs before and after, and they had 2 distinct groups. They had a morning and evening supplementation group, which is your More and Eve group here, and then you've also got a pre and post workout supplementation group. So you're trying to maximize the availability of the amino acids around the actual exercise bout itself, because there seems to be some evidence to suggest that that would be optimal. So you can see pre-post is in reference to the resistance exercise that's been completed over 10 weeks, and then you've got the morning and evening approach where you're dissociating the feeding away from the exercise bouts and you're just placing it within the 24-hour window itself. Now, what do we see in this particular figure? Well, it seems that for this particular study, the cross-sectional area of the muscle over a 10-week training bout demonstrated a significant increase in the amount of cross-sectional area of specific muscle fibers here when the supplement was consumed around the training bout, as particularly in comparison to the morning and evening where there was a dissociation. So they didn't have a control group here, which is a bit of a shame, but I would say that so there's one observation anyway. Here's another study here. Here's the same study, but looking at the amount of kilograms of lean body mass, fat mass, and body fat percentage that these represent. So you can see the lean body mass here significantly increased by around sort of 2.5 to nearly 3 kilos within the pre-post group in comparison to the morning and evening group. And similarly, their fat mass decreased very small, but these are not meaningful differences. But the percentage body fat, because they've had such an increase in lean body mass here, had an influence on their percentage body fat. Because you now have more lean mass available to kind of put that into the equation. And you can see here that the pre-post reduced body fat percentage as well. So associating the feeding with the exercise bout seems to have a beneficial impact. Now, this was another study done by Hoffman with 33 resistance trained individuals. It was 10 weeks pre and post workout versus morning and evening, very much like the Cribb and Hayes one. 4 days per week with DEXA before and after in order to give this body composition data, with a 40-gram protein supplement with 2 grams of carbohydrate as well, for probably just for a little bit of taste. And we get the same kind of picture. We get a completely different picture here. We can see that the dissociated feeding here, AM and PM, resulted in a higher lean body mass. Similarly, that had an impact on their fat-free mass as well. decrease in their fat mass, and then this would be their percentage body fat, and you can see here that this had a profound different impact in comparison to the Cribbs and Hayes. So there's general consensus then that there is an anabolic window, but what's generally not agreed upon is the duration of this effective window, because all these two studies are doing is looking at the proximity, but only within a kind of 12-hour period. You're still getting, clearly based on this Hoffman publication here, you're still getting benefit from consuming a higher amount of protein in combination with structured resistance exercise, but it doesn't necessarily have to be pinned exactly to the exercise by itself, as long as it's consumed within a given time period of the exercise itself. And that's typically, if you're increasing your protein consumption over the day's period, and you're taking into account the optimal dosing, in terms of the 20 grams as being a minimum, and you're trying to structure in the patterning, then the timing does kind of become a little bit irrelevant, assuming you've got those other two things sort of all pointed out and figured out. But it's not as important as people are sometimes make reference to, especially the Starship Trooper lad who's saying his workout is wasted. Well, not really. And I've heard some professors say anywhere sort of 24 hours to 48 hours, the muscle is still receptive to increasing its synthetic response. But there's a lot of work being done in this area to try and unpick that particular question to find out more. The final things that we're going to talk about is co-ingestion, and I'm not going to dwell on this too much, but there doesn't seem to be too much evidence to suggest that co-ingestion with carbohydrates improves muscle protein synthesis to any amount. And you can see here by this particular study here, I think the reference is washed out again, apologies, but you can see here that when protein and carbohydrates were consumed post-exercise, there was no difference between the protein and carbohydrate, whether you were exercising or at rest. But where they did find a difference, and what this meta-analysis shows here by Louise Burke, is that there does seem to be an increase in the amount of muscle glycogen that's being synthesized in the post-exercise period, which means that if you co-ingest protein and carbohydrates, that there might be some benefit for endurance athletes who are looking to resynthesize their muscle glycogen. There seems to be a relationship between the amount of carbohydrate that you consume co-ingested with protein resynthesizing muscle glycogen at a faster rate, and it could be attributed to an increase in insulin, or how receptive the muscle is to resynthesizing or absorbing macronutrients, but again, it's currently unknown. Now, fat is an interesting one. There was a nice study done by Elliott et al. Where they were looking at there was a nice study where they were looking at fat-free milk versus full-fat milk versus a calorie-matched, a calorie-matched beverage, and you can see, you can see here that there does seem to be a significant impact of consuming full-fat milk in comparison to fat-free milk. As to why this is, we're not currently sure. I don't know. I've not read around this area for a little bit of time now. But there does seem to be some impact for the co-ingestion of fat alongside protein. And again, there is lots of people working on that particular area as it stands. Now, just to add a little bit of, we're now moving on to the source. Okay, so co-ingestion doesn't seem to be too much of a benefit unless you're trying to resynthesize glycogen, but there does seem to be a role for fat to play, but we're not quite sure at this particular moment in time exactly what that role is. Now, if we then move on to the source, when we talk about kind of animal protein and plant-based proteins, you can see here that we've got some amino acid profiles for different foods that have been consumed. So you can see up here we've got skim milk, we've got soy milk, we've got beef steak, eggs, and liquid meal. And you can see that the branched chain amino acids, the essential amino acids, the TAAs, and the leucine here, they all represent different amino acid profiles depending on exactly what's been consumed. Now, skim milk here always represents the highest amino acid concentration at any given time point, but a fairly rapid drop away in every example here. Whereas your things like beef steak, if we look at what would be a good example here, if we look at our branched chain amino acids here, we can see the solid squares take longer to rise, they don't reach as high a peak, and then they fade away over a longer period of time. So things different, altering the protein source alters the amino acid concentration at any given time, and therefore that does have a downstream impact in the amount of the protein synthetic response that you're going to get. I'm actually going to skip this slide because it says everything that I wanted to say as it did before. So let's think about those sources in terms of different commonly consumed nutritional supplements, and then I think we're going to have to wrap it up because we're going to run out of time otherwise. So we've got whey protein here. Whey is a byproduct of the milk industry, and it is a complete protein source, but it just so happens to have a particular high leucine content, and leucine is a particularly anabolic amino acids. So we've got different consumptions here. So we've got rest versus exercise in white and black, and we've got whey versus casein versus soy. We can see here that we've got a significant increase in all but the casein in comparison to rest and exercise, but the whey protein is significantly increased over the casein and significantly different from the soy. So in terms of different amino acid profiles, the casein is kind of known as a slow releasing protein over a given amount of time. Soy is a complete plant-based protein, and whey is kind of like an optimized leucine source, but you can see here that it is resulting in a higher fractional synthetic rate, but the soy isn't too far behind. If we think about casein versus whey, we can see here again over longer periods of time they seem to be equal with one another because of the amino acid profile that you can sustain for a longer period of time. And also if you see the first 3 hours versus the second 3 hours, we've got different profiles there. I'm sorry, I'm rushing this a bit, guys. I would usually open up a little bit more, but I'm just conscious of time. And then we've got the soy and the whey comparison there. So I'm just going to switch views, just drop in on the chat, and then I'm going to come up with some concluding points in the background, if that's okay, guys, just because I'm acutely aware that we've politely run out of time. There is a study that I was going to refer to, which I might just touch on very briefly. Okay, we'll conclude on that slide. There is more content there than I realized, so my apologies for that. So Leona's coming in with a question. The anabolic window is that for like after a workout people have carbs or protein shakes within 30 minutes? Yeah. So when I was talking about those distinct studies where they dissociated the AM PM feeding versus the pre and post training, that's exactly what the researchers were trying to do. They were trying to associate the feeding stimulus around the exercise specifically. If you dissociate it by putting it in the morning and in the evening, and the training takes place at some point in the afternoon, then if you have two distinct groups, then you can make that comparison between the two groups. So again, it seems to be most practical to ensure that you do have some form of feeding some form of feeding just after the exercise bout. Now when people feed carbs after their exercise bout, it depends on what they're trying to achieve, but most endurance athletes would want to be consuming some amount of carbohydrates post exercise so they can resynthesize their glycogen stores, but we're not talking about that today because we're just talking about proteins specifically. If there aren't any other questions, I am going to politely come up to a summary slide here. So 20 grams of ingesting protein. So there was a study that I didn't show you, which is why this is kind of difficult. So I'm going to talk about that and then we'll close it up. I apologize for running on a little bit. All of the studies that I've shown you so far use either a single or double leg exercise. So what that essentially means in terms of a whole body perspective, it doesn't utilize all of the muscle mass that you have available to you. And as a result, you're potentially missing out on the opportunity to kind of drive that response because you're not utilizing all the muscle mass you have available. So when you do whole body resistance exercise, there does seem to be some evidence to suggest that that protein synthetic response is actually higher when you feed more protein. And I'm just going to very quickly show you that so that these concluding points make sense. Let's just head back now. Yeah, so here's some evidence that shows that. So what we've got here are individuals that either have a low lean body mass or a high lean body mass, and that's kind of irrelevant because there doesn't seem to be any difference between the 20 and the 40 grams in this particular study. But what the biggest thing is when you pull these two groups of individuals together and you look at whether 20 and 40 grams of protein post exercise has any difference. This was the first study to show that 40 grams seemed to have a significant increase in the fractional synthetic response, or the amount of protein that's being synthesized. But this was also the first study to actually undertake resistance exercise in a whole body way. So they did both lower and upper body exercise, and then they did the muscle biopsies from the thigh, or the vastus lateralis on the mid, or the outside of the thigh, which is where these biopsies usually take place. So it does seem that if you do whole body resistance exercise, then there is maybe some evidence to suggest that you could increase your protein dose above the 20 grams suggested. And therefore it kind of opens up that kind of question, well, is 20 grams optimal for everybody under all circumstances? And this is where the research is kind of going in this direction now, where the researchers are trying to kind of tease out exactly what is optimal under every circumstance for every type of person. Because again, all of the studies that I've shown you today were all done in resistance trained males. And therefore that opens up another question as to whether this is appropriate for all types of people undertaking these particular exercise bouts. So let me just flick through to those concluding remarks. So it seems that 20 grams of ingested protein doesn't necessarily maximally stimulate response for everyone, particularly in young adults, but it does seem to get you most of the way there. So 20 grams seems to be a good amount. The amount of muscle exercise does alter that particular response, and therefore further studies are going to try and directly investigate whether that actually has an impact or not. So I'm going to kind of politely close it off there. We'll head across to the chat function. So just to kind of clear our heads and just get it all cleared up. So when we think about the dose, 20 grams seems to be optimal for most people. But if you're undertaking resistance exercise that's whole body, then there is some limited evidence to suggest that more than 20 could be beneficial. When we think about patterning, we want that 20 grams to be the minimal threshold. So if you have 4 or 5 different opportunities to feed over a 24-hour period, that would then maximize your protein synthetic response over that period of time. When we think about the co-ingestion, there doesn't seem to be much evidence to suggest that you get an increase in muscle protein synthesis when you co-ingest with anything other than fat, and it seems to be whole milk that seems to be having that particular response. But we haven't really yet teased out exactly, well, what's the ratio between fat and protein that needs to be consumed to maximize that particular bout. The timing seems to be important, kind of, but only really over a 24-hour period. As long as you feed the amount that you want to consume in that 24-hour period that contains some form of resistance exercise, then the likelihood that you've optimized as much as possible. If you're not meeting the dose or the amount that you need to consume over the 24-hour period, that's going to be more detrimental than when you actually consume it. But there is some evidence to suggest that feeding around the exercise bout itself can be optimal in order to recover from that exercise bout itself. The source is important. If you're a vegetarian or vegan and you're not consuming a complete protein amino acid profile, then that's going to be detrimental to you adapting and having all the amino acids available to you in order to accrue functional mass later down the line. I'm going to sort of open it up to any further and final questions, but other than that, hopefully you found that interesting, exciting. It makes you think more comprehensively about that relationship between interventions and what what nutritional interventions may work, and the importance that the two and how they interplay with each other, and how that might actually influence and change the way that a person responds to a given intervention that you've provided. So thank you very much for listening. If anybody wants to jump off, that's absolutely fine. And if anybody has any final remaining questions, I would be more than happy to answer them in the chat right now. While I'm waiting for that, for the people on my nutrition metabolism, nutrient metabolism, I'd recommend you go and have a leg stretcher for 20 minutes or so, because we'll be back at 3:00 and we've got a seminar task which we'll do in our collaborate room to talk more comprehensively about that, and go from there. I've got the chat open on my screen, guys, so if there's... Okay, I think I've answered that question. And I'm just going to just double check who's watching. Excellent. So, 22 viewers. Fantastic. Iba, thank you very much. Mihaela, thank you, Mike. No worries. Thank you very much for joining me. That's excellent. Jessica, thanks very much for joining today. Hope you got something from it. Debbie, thanks a lot. And Leona, excellent. Thanks very much for joining today. Really good to have you on board. Sophie, great to have you in the chat. I'll see you at 3, hopefully. And Camelia, thank you very much for joining. Connor, thanks again for joining this week. Great to have you on. Sorry, I'm not ignoring you guys. I'm just getting everything sorted out in the background. We have Justin. Excellent. Thanks very much for joining today, Justin, and hopefully you got something from the thing. Cheryl, terrifying having you in the chat today. Thanks very much for joining and to giving us some questions. So thanks very much for that. And lay off the Easter egg, man. That's it's criminal behavior. Alright guys, if there's no questions coming through, I will politely call it a day and go from there. Alright guys, thank you very much for watching today. Feel free to reach out in an email if there is anything that sort of springs to mind. For those on nutrient metabolism, I will see you at 3 o'clock. Thank you very much for listening. Take it easy. I'll see you later.